Our second speaker this week is Dr. Srinivas Ramachandran. Dr. Ramachandran did his PhD with Nikolai Dokolian at the University of North Carolina in Chapel Hill. And during graduate school, a majority of his work was on the recognition of platinum DNA adducts and on modeling calcium ion channels. Thereafter, he joined the lab of Steve Henikoff at Fred Hutch. And here he developed computational and experimental methods to map nucleosome structure and dynamics in vivo. Dr. Ramachandran started his lab not too long ago at the University of Colorado, and very excitingly, earlier this week, he was named a Pew Stewart Scholar. In his lab, he continues to develop new methods to map chromatin structure at a higher temporal and spatial resolution. The Ramachandran lab is looking for interested graduate students and postdocs. So for those of you who are interested in his work, do reach out to him, or better yet, stay back for the coffee chat today, and you can find out more about what's cooking in his new lab. With that, I will let Srinivas do the talking now.
Awesome. Thanks a lot, Pravrata. Can you hear me? Yeah, okay. Thanks a lot for the organizers. This is an amazing series. I'm really happy and honored to be here as part of this. And also thanks to the great talk by Henry. He has introduced enhancers really well. So my job is going to be much easier. So this is the title of the talk that I gave, but pretty much what we are doing in this project is to ask how proteins bind enhancers and how we can understand enhancer function based on how they bind. This project was possible due to two great collaborators. First is Satya, who is a postdoc in the lab. And then as we were going to the end of this project, we got like really amazing data sets for validation from our collaborator, Kami Ahmed, who is a PI at Fred Hutch. So this audience needs no introduction that transcription factors are really critical at many different scales for organismal fitness and also for survival. So at the scale of chromosomes, we know transcription factors that organize the genome. At the scale of cells, we know that transcription factors help in response to external stimuli. And at the level of the whole organism, we know that cascades of transcription factors are essential for development. So in this multi-scale sense of things, we can think of transcription factors as these really small molecules in the scale of the organism that have cascading effects that go all the way from the chromosome all the way to the whole organism, as seen here in this nice visualization. So, however, transcription factors have to find their binding sites in the context of nucleosomes. And the question we are really interested in is how transcription factors find their functional binding sites in cells in the presence of nucleosomes, which is like a very broad and fundamental question that has been pursued for many decades now. So the angle that I want to talk to you today about is how multiple transcription factors could work together to achieve specificity. And this question also has been like worked on for many decades. And I just want to give you a taste of a couple of models where transcription factor combinations could provide specificity and kind of work on the nucleosomal template. So the first one is where you can have transcription factors that bind nucleosomes, so they can identify sites even on closed chromatin, open up these regions so that other transcription factors can bind in the presence or after the activity of this first pioneering transcription factor. And in this case, you need function of multiple factors to happen together for specificity to happen. And the other case, which is really interesting, is like how transcription factors have evolved protein-protein interactions between them, or the functional output of factor binding happens only when they are bound in a very tight orientation on the DNA. So the factor binding sites have to be positioned at very specific locations relative to each other. So throughout these two kind of, for me, are in extreme cases. There is a whole continuum of function that's possible in between, where you have multiple factor binding sites, and factors can either bind independently or at the same time to ensure that those sites are specifically recognized in the cell. So we wanted to ask how transcription factors might bind at functional sites. And as you saw from our previous talk, enhancers are a perfect system to study this because they usually are driving gene expression a lot of time in a cell type specific or in a temporally tight windows during development. So when we study transcription factor binding in enhancers, we are automatically limiting ourselves to functional binding sites. And so the question that started this whole project was very simple. How can we characterize factor binding at really high resolution at functional enhancers? And the system we are working on today, like we'll be talking about, is Drosophila S2 cells. And that's really special because we have a map of functional enhancers in these cells due to this really nice method called star-seq, where DNA fragments from the whole genome are cloned downstream of a transcription start site in a reporter in an intron. So it's very similar to the intronic enhancer we heard from in the last talk. And if this sequence has any enhancer properties, it's going to drive the expression of the promoter in front of it. And it'll give rise to an RNA which contains the enhancer sequence itself. So just by sequencing this RNA, we can get a map of all the active enhancers, all the sequences that can act as enhancers in that cell type. And what's really cool about this technique is that this actually happens on a plasmid. So it doesn't have the endogenous chromatin context. So when now we go back to, so there are like around 6,000 enhancers mapped genome-wide in S2 cells, which we are working with. And when we go back to look at these enhancers in the endogenous chromatin context, we can come up with really interesting things. So the reporter itself in the plasmid context can have very high activity, but it can exist either in open or closed chromatin in the chromosomal context itself. This was shown in the original paper, and we could reproduce this beautifully, where you can take all these enhancers, they are like around 500 base pairs in width, and then cluster them into two groups based on their accessibility here, H3, K27 acetylation, and DNase. And when we plot chromatin modifications that's associated with silent chromatin, which is H3, K27 trimethyl, it basically maps in the opposite way. So we basically have two sets of sequences, both of which have the capability of being enhancers, but only one set is an enhancer in the cell in the native chromatin context. So we can really ask how transcription factors bind and distinguish these sites. So to ask how transcription factors might bind these sites, we wanted to have a very general method where we look at transcription factor binding at really high resolution. So we turn to micrococcal nuclease. And if you don't know about MNase, I'm just going to give you a really short sales pitch. So you basically treat intact nuclei with nuclease. You're going to get protein DNA complexes that are left behind after the MNase chews through most of the accessible DNA. And you just sequence the DNA, map it back to the genome. The length of the fragment is going to tell you a lot about what kind of complexes are formed at different parts of the genome. And a striking example of this is that you can basically take active genes and average the nucleosome size fragments around 147 base pairs from all the active genes, and you immediately start seeing a nucleosome depleted region, which corresponds to the promoter, and ordered nucleosome arrays both downstream and upstream of the gene. But what's really interesting is, so this is the general way of doing MNase to look at nucleosome positions. But what we can do is we can enrich for all fragment sizes. And now if we look at only really short fragments, we get a really different picture where you get enrichment only over the promoter, where we think this is coming from transcription factor and RNA polymerase protections. and the actual distribution over the gene bodies or upstream is very, very low. And this is highly specific because if we look at non-expressed genes, we won't see this enrichment of these short fragments. So we turned to these short fragments to basically understand transcription factor binding. And we did a small trick where we just ran out the MNase products on a polyacrylamide gel and cut out the gel that corresponds to 100 base pairs and smaller. So we are now highly enriching for these protections that would correspond to subnucleosomes and transcription factors. And now when we take these and map them back to the enhancers, what we see is it basically matches really well with known mediators of accessibility, which would be like high DNase hypersensitivity signal or high K27 acetylation. But the added advantage now is this is like a low resolution view of this data set. We could go at base pair resolution and start seeing a lot of features. So first, I'm just going to show you a closed enhancer. So in gray is the nucleosome profile, which will be around 150 base pairs. And red is your less than 50 base pair fragment centers mapped at base pair resolution. And what we can see is that we don't have much enrichment of these fragments in closed enhancers, as we saw in the last heat map. But if you look at an open enhancer, you get really sharp peak with this like 30 base pairs or even lesser, which basically is telling you specific transcription factor binding within the enhancer. So these orange lines are basically the edges of the enhancers as defined by star C. So what you can see here is the nucleosomes are more arranged, and you see a depletion around the transcription factor binding site as defined by these short fragments. But this is not the only thing we see. We actually see multiple transcription factor binding in enhancers. So here is an example where we have 4 binding events, each with different strengths. And again, you see that the nucleosomes are kind of moving away from these binding events. And so the width of the nucleosome positions is actually proportional to how many peaks you have in between. So in this enhancer, you see 4 peaks, and this is actually more like the average behavior, because when we count the number of peaks within these orange lines for all active enhancers, we actually see a nice Gaussian distribution with the mean around 4 peaks. So we can actually have a way to study transcription factor binding at really high resolution at enhancers with multiple binding events happening at the same time. And this can now let us ask this really important question of how do these transcription factors actually bind enhancers cooperatively. And when I say cooperatively, it is like the examples shown in the introduction, where binding of one transcription factor is predictive of binding of the other. Or in other words, you see both two factors bound at the same time compared to either one bound individually. So to ask this question, we first wanted to simplify this data, because we have like many different combinations of transcription factor pairs possible. So we want to first just look at the strongest possible pairs. So to do this, we just took all the open enhancers and aligned them at the strongest peak of these short MNase fragments. So you see this here, it's really nice, and like very small fraction have a weak peak, but most of them have a really strong peak. And then when you cluster these profiles, what you see is that you basically have each cluster having a strong secondary peak, which is at a different distance from this main peak. So of course, the cluster 3 is a nice control where you have one strong peak, and this would be representative of one strong transcription factor binding event driving the enhancer. But all of the other clusters have at least 2 strong peaks, and they are basically divided up into positions, into clusters based on the distance of that secondary peak from the primary peak. So to ask if these are actually forming transcription factor binding in an orthogonal way, we turn to V-plots. So V-plots basically make use of all fragment sizes to infer particle binding, And here, this is just a simulation where if you just have a protection in the center where you cannot have MNase cut, and MNase has basically it's randomly cutting throughout this region. You plot the position of each of the fragments that generated on the x-axis and the length of the fragment on the y-axis. You automatically get this pattern, which is basically telling you that you have, just as a result of this protection, you're going to have depletion of the fragments in this region around the protection, giving you a shape of a V. So wherever you see a V, you can infer what is the size of the protection and where the protection is actually present. So we can basically take the third cluster I showed you before, which is basically the cluster with one major transcription factor binding event according to the short fragments, and really ask if this represents a strong transcription factor binding based on a V plot. So we take this cluster and make a V plot, and what we see is just like the simulation I showed you before. You see a really nice V and a depletion of fragments around the V, which would say that there is a nice 40 base pair protection that's driving this peak. And we can confirm that by just plotting the we can take a slice of the heat map right in the middle and basically plot the density of fragments as a function of length. So you can see that you have a really nice single peak, which should correspond to the single protection right in the middle. So the advantage of V-plot goes beyond just confirming that there is a single particle bound. The real reason we wanted to do V-plots was to see if we can detect two particles bound at the same time. So we asked the question if we can analyze protein co-binding using V-plots. So to do this, we turn to the fourth cluster where you see there are two peaks that are very close to each other. And then when we make a V-plot there, what we immediately see is that you have a first major V that you see similar to the cluster 3, which represents the central peak. And then you have a weaker V on the side, which would represent the secondary peak. But really interestingly, you start seeing this third group of enrichment, which corresponds to a third V, which you can kind of see here. And we used only less than 50 base pair fragments to call these peaks. So we basically wouldn't even have known the presence of this third peak when we did the analysis. This is coming out when we build the V-plot. So now, if we were to think about all the V's we can kind of infer from this graph, we basically get the three V's as I told you about, and we can infer that the first V is coming from a specific protection that's at the center. The second V is a protection at around 40 base pairs from the center. And the third V is basically when both the proteins are bound at the same time. And we can confirm this by just taking slices of the V plots. If I take a slice right here in the middle, we get a nice peak that corresponds to a single particle binding. And if we take a slice in the middle, around 20 base pairs, which will be the center of this particle, we see the second peak that corresponds to both the proteins bound at the same time. So even though MNase kind of breaks up the chromosome when we do the experiment, and you basically lose information about particles that are bound together, because of the particles being tightly bound at the same time, we also get protections. And with this kind of V-plot analysis, we are able to basically infer two proteins bound at the same time at enhancers. And we can basically move this second V to the right and ask if we get corresponding protections that would, if we get protections that correspond to this co-bound species, and that's what we did. So we can move down these clusters. So you can go to the cluster 5, which is like slightly more separated. So your V's are, so the V's are slightly farther apart. And then you get this extra density right here, which is exactly the length at which we expect co-bound species. And we can go to the next cluster. And here you actually move on to larger fragment sizes where you have two positions that are around 80 base pairs apart. And when these two transcription factors are bound together, you get this third V really nicely telling you that these factors are bound at the same time. So what we can do is to ask if this is actually a significant density, we can calculate how many fragments we expect at this position for each cluster, and normalize that to the cluster 3, which has only a single transcription factor bound, to kind of get the excess co-binding that we observe in these V plots. So when you do this, so of course cluster 3 you set it as 1 because there's a single protein binding, it gets normalized out, but you get 4 to 6-fold more co-bound species compared to single binding, which basically, this and other simulations I'm not going to now, but I can talk about later, basically reveals that we have basically substantial and widespread TF cooperativity at active enhancers. Of course, this is basically mainly inferred from these V plots and MNase analysis. So we wanted to confirm if these protein binding events that we see at enhancers can be seen by other methods as well. So we basically looked at this really cool method called dual enzyme single molecule footprinting, which was published by Arnold Krebs in 2017. Like really nice for us, the experiment was done in the same cell line that we are doing. So we basically turned to this method to confirm protein binding in enhancers independent of MNase. And the way this method works is basically takes advantage of the fact that Drosophila doesn't have much DNA methylation. So we can take bacterial methyl transferases that can methylate the CpG or GPC context and treat Drosophila nuclei with these methyl transferases. When transcription factors are bound, then these methyl transferases do not convert them, do not methylate them. So you basically have these cytosines unmethylated. Then you can extract the DNA and do bisulfite conversion. And the ones that are basically protected are going to be different from the exposed cytosines. So then with the sequence data from this bisulfite conversions, we can basically take any unmethylated cytosine surrounded by methylated cytosine to represent a protein bound footprint in the genome. So we did this analysis on our enhancers. And to start with, we wanted to make sure that there is no sequence bias in calling footprints, because this, of course, depends on GPC and CpG context in the enhancers itself, and enhancers can be pretty short sequences. So first, what we did was we called all theoretical footprints just on DNA computationally, and then we basically see that the footprint sizes are pretty much the same between closed and open enhancers that we defined earlier in the talk. But then, if you look at the data from S2 cells where we call the footprints, we basically see first, really interestingly, you have two peaks, one short footprint peak and one long footprint peak, which really nicely corresponds to transcription factor binding and nucleosome binding. And we see that open enhancers have more transcription factor footprints and closed enhancers have more nucleosomal footprints. So overall, like basically, if you had to add another track here, you're going to see that the DSMF method basically captures the same thing that was captured by DNase and MNase. So this now gives us the confidence that we can basically look at transcription factor binding in much longer fragments than MNase. So you basically have around 270 to 300 base pair fragments where we can ask how transcription factors and nucleosomes are binding relative to each other on the same fragment. So the main question we wanted to ask was, of course, like can DSMF reveal the co-binding that we saw in the V plots? And to ask this, we basically took these methylated fragments and just showing you some like real data compared to the cartoon I showed you before. And we can basically call all different states of the enhancer. So here, the gray box basically represents the transcription factor binding site. And this line below is the whole fragment we get from the bisulfite sequencing. And you can have fragments where you have all the CPGs or GPCs methylated. So this would basically represent a state of the enhancer where it's completely exposed. Or you could have basically only the CPG or GPC within the transcription factor binding sites unmethylated. So we call a nice footprint that encompasses the binding site as defined by the MNase-seq. And the other option we see is basically where you have these longer footprints that cover the transcription factor binding site, and the length of these footprints make us infer that this is a nucleosome covering the transcription factor site. So for a given enhancer, we can basically map out all structures and all states with this data set. Now, if you move on to a state enhancer with two peaks, basically can start seeing footprints that indicate that both the binding sites were occupied by transcription factors. So basically, having an unmethylated cytosines in both the binding sites at the same time is telling us that in this enhancer, in this particular molecule coming from the S2 cells, you basically have both the factors bound at the same time. And we can actually go on, we can look at three sites. So here in the same enhancer, we basically saw that these are the two peaks we were looking at, but there's a third one also where you can have three factors bound at the same time. So if you have three states for one binding site, then if you have two binding sites, you have 9 states, 3, you have 27, etc. So just to kind of have enough coverage, we stuck to 2 binding sites at a time, but you could actually look at multiple sites at the same time for sure. So when we do this, we basically take MNase-seq. So here I'm showing you a single enhancer where you have 2 transcription factor binding sites, and then you take all the reads and categorize them into different states. So first, you could have no factors bound at either of these, so they are completely exposed. Or you could have factors bound only on one of the sides. So you have this site bound or this site bound. So this will be like single binding event. Or you could have both of them bound at the same time, which would be your co-binding event. And if this co-binding event is happening at much higher frequency than you would expect based on the single binding event, then it tells you that there is cooperative binding in these enhancers. And you get the complete picture because you also get the events where you have transcription factor bound on one side and nucleosome on the other, or the enhancer is completely occluded. So since you get both the states where the enhancer is completely exposed, which you wouldn't get by MNase because there are no protections here, and states where they're completely covered by nucleosome, we basically get a complete picture for this pair of binding sites. And this enables us to basically ask to what extent you have cooperativity at any 2 pairs of transcription factor binding sites at active enhancers. So we basically can set the expectation for any state for a pair of the states basically based on the probability of the individual state. So I'm just going to go back to kind of walk through that again. So based on one factor bound, you can basically estimate what is the expectation for both of these to be bound at the same time. So this is going to be a ground probability. So across all pairs of binding sites in the active enhancers, this is the ground state probability we have for 2 nucleosomes to cover both the binding sites. But what we actually see is that the observed probability is higher than what you would expect based on individual probabilities. And this is true for all homotypic combinations. So if you have both covered by nucleosomes or both exposed, or both sites bound by transcription factors, there is significantly higher probability of observing them compared to what you expect based on their individual probabilities. And the case is exactly the opposite when you look at heterotypic interactions. So when you have either one site is bound by transcription factor and the other is exposed, or one is exposed and one is nucleosome, and one is bound by transcription factor and the other is bound by nucleosome, these are actually depleted compared to what you expect. So the fact that homotypic states are overrepresented points to the like very strong possibility that all the transitions of pairs of sites are happening cooperatively, because you see more of both states being the same rather than them being different. So we decided to focus on the state where both of them are bound by transcription factors, as these would be answering the questions we asked before regarding cooperativity. And we can basically, based on these two probabilities, set a score for cooperativity. And that's what I'm showing here. So this score goes from 0 to 100. Basically, it's a normalized extent of co-binding. And we can set a probability of observing these events. So it's which is plotted on the y-axis, and it's a negative log 10. So higher the value, the lower the expectation based on individual binding. And what we see after adjusting for multiple testing is that 31% of pairs of binding sites have highly cooperative binding, which is not what you would expect based on looking at 5,000 sites that are at very, very different distances. And what you can ask is that each enhancer can have multiple pairs, because as I showed you before, there are on average 4 binding events per enhancer. So if you collapse these pairs into enhancers, what you see is actually really striking. So if you have at least one pair of transcription factors that are binding cooperatively, you basically capture 64% of the enhancers. And 28% of the enhancers have actually more than one pair of transcription factors binding cooperatively. So, which confirms what we observed before from the V-plots, that the cooperative binding is basically very widespread and happening with quite a lot of strength. And what's really interesting is that if I take all the ones that are significant and ask what distances are they separated by, the ones that are the strongest, so this is the 76 to 100, are the ones that are around 40 to 50 base pairs apart from each other. And the weakest ones that would fall mostly below this p-value cutoff are actually the ones that are farther away. So most enhancers have closely spaced sites that are binding factors cooperatively. And Satya and I were looking at this data, and he basically showed me this paper, classic paper I had actually forgotten about, which is really helped us think about what this data is telling us. And this is from the John Widham's lab. And what we saw in this paper was basically very similar to what we are seeing in the enhancers in Drosophila. So what they did was they basically had this nucleosome sequence where they are binding completely unrelated factors. So they are binding Lexay and different enzymes that will cut the DNA if they're exposed, but they need to bind the DNA first. And what they saw was that if the factors are separated by less than 70 base pairs, so they're on the same side of the nucleosome, they have a really high cooperative binding, whereas if they're farther away, they don't have as much cooperativity at all. So the fact that you have unrelated proteins getting cooperativity as soon as you have the context of nucleosome really tells us that this might be a chromatin process that's leading to this, what we observe. And this also matches our analysis because we are looking at factors that are separated at least by 30 base pairs. So they're really very small chance that they are directly having protein-protein interactions. To test this, we directly asked, okay, what is the correlation between nucleosome occupancy and these different states of the enhancer? So we took pairs that are bound independently and compared to the ones that are bound cooperatively. And what we see is that the pairs that are bound cooperatively have much higher nucleosome occupancy by H3 chip. So there is no, like we know that these are basically representing nucleosomes. And then we can look at nucleosome turnover And what we saw was really interesting because nucleosome turnover is also the highest for the cooperatively bound transcription factors. So this would mean that basically you have enhancers that have really high nucleosome affinity, but they're also being removed at the highest rate compared to other enhancers. The ones that have less nucleosome occupancy and they don't have much turnover are the ones that the transcription factors are just basically coming and binding like as if in a billboard, like so-called billboard model. So our like major take-home, like one take-home message you could take from this talk would be basically that if cooperativity could enable efficient nucleosome displacement at enhancers with high nucleosome affinity. So this kind of gives a model of where how chromatin could basically regulate enhancer function and also establish specificity by requiring multiple factors to bind at the same time. So to conclude, basically what I've shown you is that we've developed methods to look at two factors binding at the same time by V-plots using this dual molecule, single molecule footprinting. And basically what we see is that the enhancers with multiple sites basically feature this transition where both sites are exposed at the same time and where both sites are bound at the same time, possibly because this helps in removing nucleosomes. With that, I would like to acknowledge my lab for being great teammates. And of course, I talked about work by Satya and our collaborator Kami. So we have really cool cut and run data I didn't show here, but I'm happy to talk to about, which also confirms a lot of the findings I showed you here. And funding, and I'm happy to take questions.
Thank you, Srinivas. That was a really beautiful talk. So I'm gonna give people a couple minutes to raise their hand. And I'm gonna ask Hanukkah, who's one of the organizers, to ask her question first. Hanukkah, do you wanna ask your question?
Sure. Hi, that was a fantastic talk.
Thank you.
I was just thinking that I guess one of the main questions I have is like about what the actual factors are that are co-binding, right? And I won't ask you about the motifs, like that might be a separate story. But at the start, you mentioned that RNA pull 2 could also create some of these smaller fragments. And so I was wondering if you'd ever looked at ProSeq. And basically, do you think when you see two peaks, is it often two transcription factors, or could it be one transcription factor and one RNA pull 2, for example?
Yeah, so one of the first things, actually, it's a really good question because the genesis was around the time where eRNAs were being discovered. Like there's a lot of papers on enhancer RNA. So we are like basically looking at to what extent we'll actually see Pol2 here. So using 3' NT, which is like similar to NetSeq dataset in the same cell line. And what we actually saw was that both by Pol2 chip and 3' NT, it's not very high. And I think like it's like, especially compared to promoters, it's very, very low, which is what you might expect from eRNA papers. But also, we think when you compare the levels of transcription factor binding, so we have some data on GAGA factor chip. So the correspondence between the levels of GAGA factor binding and the GAGA factor motifs in this enhancers is much, much higher than what you see with Pol2. So Pol2 might be there, but it's going to be there for a very, very small time. And also, most of these are not, like many of these are like outside of genes also. So both the fact that you're going to have very less Pol2 and also they might not, like they have much higher levels of transcription factors would tell me most of these events are when both are transcription factors.
Thank you.
Mm-hmm.
Okay, we have a question from Mike Mears. Mike, I'm gonna allow you to talk.
Hey, can you hear me?
Yeah, hi Mike.
Hey, Srinivas, great talk. So I am going to go back to this point actually. So in your model, you would sort of infer that the cooperativity is dependent on distance between the factors and not the identity of the factors themselves. So have you looked at motifs to determine whether there's more cooperativity between certain pairs and not others?
Yeah, so the motifs are actually a beautiful story. So there are a couple of points about motifs. So one is that, yeah, if we actually ask what are the pairs, so that was the ideal thing, right? So you basically get out these rules for factors at specific distances that are highly cooperative, and we didn't find any such thing. Like there were like very little enrichment for certain pairs, but not really. But the basis for that could be actually a bit more complicated. So one of the things we noticed was that I would say around I might be wrong, but like around 40% of the sites or more had no detectable motifs by the standard cutoffs of we have for motif scanning algorithms. And this is like this is a point actually Kami brought up with me, that like Drosophila's transcription factors are really well characterized, and we are looking at all the ones that are expressed in S2 cells. So that's actually very striking. And one of the things that he has seen before is that if you actually reduce the cutoff for looking, when you scan the motifs, you actually start finding more, obviously, but it makes more sense because here we have really high resolution, like 30 base pair peaks, and we think that we have to like change the way we find motifs to actually ask what would be the factors that are binding, and whether there are like specific pairs that are overrepresented. Because the standard ways would work for a lot of sites and like maybe wider sequences, but here, since we have such high confidence that this is transcription factor bound and it's nucleosome depleted, we might go with some other methods to identify these motifs. So then we might find specific pairs.
Great. Yeah, I'm sympathetic to the motif question, obviously, because yeah, I've done some work with sort of lower affinity motifs too at these sorts of sites. But yeah. So I appreciate
but when we do take the subset that has really high affinity, we can clearly see Gaga factor is the best example. So I'm not gonna walk too much into this, but it's so we basically can take all the Gaga factor sites in our MNase, and you basically, when you look at the chip data, it matches at really high resolution, and you can basically look at co-binding very well with these in these sites, and the secondary sites are going to be very different motifs. So if it is Gaga factor and we know it interacts specifically with other factors, but you see cooperative with all types of other factors we don't know interactions with. So I think definitely there is specific interactions that are driving cooperativity for sure, but I think that there is a general picture like the Widom paper where you have completely unrelated proteins driving cooperativity. And this has been shown in yeast also, where they could space different transcription factors that are not endogenous to yeast to drive reporter expression.
Great. This looks really nice. I might follow up with you offline here so I don't take any more time, but thank you.
For sure. Yeah, thanks.
Thanks, Mike. Okay, I'm also going to unmute Matthew Wooten.
Hi, Srinivas, can you hear me?
Yes, I can. Hi.
Awesome talk. I was just wondering if developmental versus housekeeping enhancers cluster differently on your data set depending on spacing or potential for cooperativity.
Yeah, that is an amazing question. We have thought about that. I think that's the next thing to do. So we basically, the questions we want to ask are like how does it look at developmental enhancers versus the housekeeping ones, how it looks at promoters, and how we can like incorporate different transcription factors that will also be kind of telling you about like more cell type specific versus broad functions. So yeah, so that's something we want to look at for sure.
Well, that sounds very exciting.
Thanks.
Yeah.
Okay, I'm going to ask you some questions from the Q&A. We've got a lot of questions that we may or may not cover today, but Joaquina de Las asks, I'm curious about the defined sizes you find between small MNase peaks, presumably transcription factor binding events. Is your data suggesting that the factors are always at defined distances, or were you just showing us a subset of specific sites?
Yeah, so this actually brings a really important point of contrast between the single molecule footprinting and the V plots, right? So for us to see really good signal in V plots, what we did was to like cluster sites that were very similar to each other, cluster enhancers that are similar to each other. So actually going back to the clusters, it actually makes sense that you have, so quickly, almost, yep, there you go. Yeah, so I don't know what the null expectation is for the distances. So it is very striking that we're able to cluster this at these distances very nicely. But ideally, if we knew the null expectation, we could say if this is like more than what you would expect by chance. But the advantage of doing this separation is that now we can look at enhancers with very similar features. So the short answer is that we really don't know if specific distances are preferred. It seems like there is a peak at 40 or 50 base pairs for all pairs of peaks. But since we haven't modeled the null expectations fully, we don't know if that's expected. But this became really useful to us to generate the V-plots because we can classify enhancers.
Okay, we do have a lot of questions, but unfortunately we're running out of time. So I'm going to encourage people to please join us for the coffee chat afterward. We've already sent a message on the chat box, so just reply there to tell us that you want to stay. For those of you who are not staying for the coffee chat, thank you for coming to the seminar again, and we hope to see you next week, and maybe you can stay for the coffee chat then.